Using an automatic learning approach based on genetic data, a trio of scientists from Scripps Research has identified a CFTR protein region, which is vital for its appropriate function, but is barely affected by existing cystic fibrosis treatments. The researchers described their results in the study, the triangulating variation of the population, to define the mechanisms of precision management of genetic diseases, the structure of the journal. The human genome contains thousands of genes that provide instructions for making proteins. The code of each individual is unique and, therefore, there are many variations in the proteins produced. Sometimes these variations cause problems. For example, mutations in the CFTR gene can cause FC, but other variations have more subtle effects on the structure of proteins and functioning. Here, the researchers have designed a new automatic learning approach called variation, or variation capture map, which uses advanced statistical modeling to make predictions on how the variations in the genetic code are likely to result in differences in protein structure, and, therefore, protein function. Recommended reading: reducing the dose of Trikafta can alleviate mental side effects, study reports. VARC provides a new distinct approach to algorithms which predict only structures, because VARC, as a tool based on the genome, captures structural relationships in the context of the native function of the protein folded by the fact physics, chemical and/or cellular cell properties found in the evolution of the human population, wrote researchers. The algorithm is designed in the levels of increasing complexity, first determining how individual genetic variations are likely to influence protein, then accumulate to grasp how the variations are displayed and how they would affect interactions with other molecules such as drugs. When you want to treat patients, you must really appreciate that different therapies could target different variants in a completely different way. And that is why our approach that examines many different variants is so powerful, said Chao Wang, a scientific staff at Scripps and study co-author, said in a press release. Our approach not only reveals how these variants contribute to the biology of each patient, but also links them in a way that each variant can inform the management of others, added Wang. In particular, the algorithm requires only a few genetic sequences to make these predictions. The fact that we can get